Hello students, this is Anusha Suri. I'm your biochemistry faculty. And hello, how are you? What are you doing? And I hope you all having a good evening. Okay, children, now it's like exam time for all of us, right? So I think some fear or something would have been in our heads around. So to avoid to get you people free, here we are to present some of our ideas and let us discuss at some of the concepts and let us make some things easier so that your biochemistry makes easy for you people, right? Yes. So what else? How I hope how your preparations are going on. I think it would be tough for you, right? I know, I understand biochemistry is little complicated, but when you just start practicing it, preparing it, it's definitely a easiest one, right? So we are here to make you this biochemistry easier, lively, and topics easier for you. And don't forget to subscribe our channel Prakara, right? YouTube channel down. The link is there. So children, for us, the complicated topics in biochemistry, first when you think of around, we think, oh, carbohydrates, structures are too tough to remember, to memorize, to practice, right? But once the things are easier, when we know the tricks behind it, definitely we feel, yes, yes, we can do it, right? So yes, children, yes, students. So I hope some you for you people certain topics like hemoglobin structure, we know porphyrin ring structures. It is so complicated. It's renal test, LFT test, how to remember these values, right? Somewhere we have in our head the complications around. So let us make it easier by clearing our doubts, by by knowing the easiest ways. And if we know what is the content, what we have to read, and like what is the important chapter, what is the important what is the weightage for it. Okay, these questions are very much important. They have been repeated since for many question papers, and then we feel yes, these are for sure questions. And when we practice them and when we go through it, then we feel yes, yes, it is very much easier for us, right? So here in this session, we will definitely have a complete discussion onto it. We will have a complete idea and a complete knowledge. Okay, these questions can be asked. Okay, in this way we are supposed to answer. Then we'll definitely feel, okay, this is what is the biochemistry. This is nothing but rocket science, right? So within a few minutes, we'll just go ahead with the syllabus, our content, and what all are the important questions to it. Okay. Yes. So students, for us, we really have a lot of okay, this biochemistry, is it that helpful for us? Yes, it is very much helpful for us. And we also have a thought, okay, can't we skip as a student? So let me skip this biochemistry. It will have in our heads. But once we are into this, when we get up with the structures, when we get up with the pathways, cycles, urea cycle, and all the tests, okay, this functions, organ functions, and when we learn about this, when we know the tricks behind it, we'll definitely feel yes, it is easier to remember the enzyme names. It is easier to remember all the nomenclatures that is there for the enzymes, and it is very much easier to remember the process of the carbohydrate digestion and protein digestion, fat digestion, metabolisms. Oh my God, we think it's too complicated, right? But once we understand, yes, this is the trick behind it. This is the small things that are left in our pathways which we need to remember. Then definitely it will be easier for us. So in these sessions, we'll be learning about it. Okay. So let us start off with our session. So as is applied biochemistry, if you see around, we have a list of units. We have almost 8 units, right? Unit 1, that is carbohydrates. Unit 2, it is lipids. 3, proteins. Unit 4 is clinical enzymology. Unit 5, acid-base maintenance. Unit 6, it is heme catabolism. Unit 7, organ function tests, wherein we'll be learning about the biological parameters and the normal values of it. And Unit 8, it is immunochemistry. So out of all these 8 units, I know you people would have gone through it and would have felt, okay, these structures are a little complicated and what to study, what not to study, which chapter is very much important. These questions are for sure. So these things we will be learning a lot in this session, right? So out of this 8 units, we will just come to know what is which of the units we need to focus more, we need to concentrate more. Okay, we feel yes, from this units, definitely we can get short questions. The weightage of these chapters are very much higher enough, wherein if we concentrate, we can score for our exams. Right? We are having OU exams have been starting from 3rd October and JNT or it's on its way, right? So you all should get ready for it, right? So we'll just go ahead with the next slide. We'll see the weightage and the importance. You see, what are the weightage units? First unit, that is your carbohydrates. Carbohydrates, it is a very important chapter, wherein in this chapter you will be concentrating more on the what is carbo, what are carbohydrates, what are its classifications, and what are the various types of the carbohydrates, its importance, right? And here you'll be learning about the various cycles like your glycolysis, HMP pathway, your all your different cycles you'll be learning in this chapter, right? And that's how this chapter has a lot of questions being posed, and it is a very much important chapter. Then coming to your unit 3, proteins, right? So proteins, definitely they are the building blocks of life, right? So in this chapter, chapter is also a very much important chapter and has a high weightage. Therein you'll be learning about complete structure of the proteins. And you'll be learning about the various classification of the proteins and the importance of the proteins. In unit 4, it is clinical enzymology. This is the most important chapter wherein you'll be learning about the structure, functions of all your enzymes, the classification of your enzymes, right? And yes, structure of enzymes is quite fishy and tricky, right? Yes, we feel okay, ma, it's very much difficult to remember the active site. What is this active site, right? So, what are the substrate molecules? So, but yet, this chapter it gives more weightage, and you will be more questions are being posed from this chapter. The next is your acid-base maintenance, then your heme catabolism, immunochemistry. So out of your 8 chapters, these 6 chapters, these complete 6 chapters, that is your unit 1 carbohydrates, unit 3 proteins, unit 4 clinical enzymology, unit 5 acid-base maintenance, unit 6 heme catabolism, unit 8 immunochemistry. So these units, they have high weightage and they definitely add up to your scores. So if we concentrate on these chapters, definitely we can score high, right? So please do subscribe to our channel. You see our first chapter, unit 1, carbohydrates. Carbohydrates, right? So first thing what we get in our mind when we talk about carbohydrates. Yes, we think, okay, today I just had my pizza. Today I just had my piece of rice. I just had piece of my bread, right? So that is what we think, carbohydrates. Today I just dumped in my burger. Today I just dumped in some sort of cold drinks. No, so carbohydrates, they are very much essential, energy givers for us. So in this unit, we'll be learning about the digestion, absorption, metabolism of carbohydrates and their related disorders. So first thing, carbohydrates means it are nothing but your polyhydroxy aldehydes or ketones, right? So that is what we get in our mind. Carbo, organic, carbon, made of carbon, hydrates, right? So hydrated water molecules. So here in this chapter, you'll be learning about the regulation of blood glucose, diabetes mellitus, type 1 and type 2, their symptoms, complications, management, how the management has to be done, the lifestyle management, and all you'll be learning in this chapter. Then investigations of diabetes mellitus, that is how your oral glucose tolerance test it has been done. What are the indications? What is the procedure behind it? And how do we interpret? And what are the various types of your glucose tolerance test curves? And what is your mini glucose tolerance test? And what are these extended tests you'll be learning in this chapter? And of course, you'll be learning about your HbA1c, hypoglycemia, definitions, and causes. So these are the important aspects which you'll be learning in this chapter, along with your classification of your carbohydrates. So we'll just see what are the important questions that are being asked in this. So if you see here the important questions, first one is definition of your carbohydrates. How do you classify them with the suitable examples? So how do you write this? When first you get this question, define carbohydrate. So in short, you can remember it has polyhydroxy, aldehyde or ketone. And here you will be completely classifying. In short, you will need to give the classification and you will give the complete chart classification chart, and then you will be classifying your carbohydrates based on the number of carbons. That is, like you'll be classifying it based on whether they are the monosaccharides, disaccharides, polysaccharides, and then you will give the respective example to it, right? So you will just give in short what is a carbohydrate definition. We need not write the pages, but the exact simple point if you're defining it, and then with the clear classification. Yes. So this is how the classification of your carbohydrates, when you mention with the definite suitable examples of your monosaccharides, then your disaccharides, then your polysaccharides, and then if you clearly classify them based on your structural as well as the storage polysaccharides. So that is all in a short way you can explain it. Then coming to your next question, that is your define oral glucose tolerance test. So what is this glucose tolerance test? Glucose tolerance test, it is like it tells us what is our capability of our body to, to as or the ability of our body to take extra glucose, right? So onto this, we are just going to give the definition of what is the glucose tolerance test, and we will be giving its indications. Indications in the sense like what are its, how do you go through, like before that test, what you are going to do. Like for example, you will eat low fat meal and you will be fasting the before day. You will be almost fasting the before night 8 to 10 hours, or at times 12 hours the fasting will be done. And then you should be very much careful with your lifestyle, right? You should not smoke, you should not have your alcohol, and you should not have a strenuous or physical activity. And also here, once your glucose tolerance test has been explained, then you will be explaining about the types. That is, types are like your glucose tolerance test. It is basically of your two types. First is your of your oral glucose tolerance test. The other one is your intravenous glucose tolerance test. Intravenously, wherein your glucose is injected for 3 minutes intravenously, and then it is being absorbed. The next is your, in case of your oral glucose tolerance test, what will happen here? The GTT level wherein the performance will, the test will be before. If you go to the biochemist, he will just give you the instructions what we are supposed to do the before day, how our lifestyle is there, how we have to maintain our lifestyle. And once it has been done, when we go for the test and when we are ready for the test, so what we will do there, you are being orally given, or you're orally administered with the 8 ounce or an 8 ounce of water, almost your 75 grams of your glucose has been given, and that also in intervals, and the blood is being drawn before, post your meal, and then after post your meal, and post your fasting, and after post your prandial also it is being taken. And then later what will happen, that all the red blood as well as the urine levels are being checked out during this test. So you will basically explain, just you will define what is your oral glucose tolerance test, you will explain the indications and its types in a short way. Then coming to your very short answers, your regulation of your blood glucose level. So first thing, what do you get it in your mind when there is a blood glucose level? So it is like we will maintenance of your glucose level in your body. Right? Normal glucose, normal random blood sugar level, it is 70 to 140 mg, right, per DL. So what happens? how it is your, what happens if your levels are increased, what happens if your levels are being decreased. So you will clearly briefly explain about your fasting sugar, postprandial sugar, as well as a random blood sugar. And in short, how it is being regulated, how the maintenance of your glucose level is being done, what is the role of your hormone, insulin hormone there, you will mention in a short way. Then you will explain, the next question is the explain the types of your GTT curves. So you will clearly give a graphical representation of your glucose tolerance curve. Here, this glucose tolerance test curve, it will clearly, in this session, what will happen, the blood and the urine samples are being collected at the various intervals. For example, for the 30 minutes, 50, 30, 60, 90, 120, like that, it has been taken around in the various intervals, and then and the concentrations of your urine as well as your blood levels are being measured, and the types of your GTT curves you will plot it. So in short, you will explain about this, right? Then coming to your lactose intolerance, the next important question, which is they have almost asked, the first two questions are the first short questions, and they have been asking for almost long, 3-4 years back question papers. I know that constantly in your question papers, they are asking this question. Then coming to your third question, that is your lactose intolerance. So the first thing that we get in our mind, what is lactose intolerance? There are certain people who are intolerant to the milk, sugar, lactose. So in this answer, you will clearly define what is lactose intolerance. That is the lactose deficiency, that is inability to digest the metabolic lactose sugar. So you will clearly define it, and then you will give the causes. What could be the underlying causes? What happens when the reason is small intestine does not make enough enzyme to break down your lactose. So since your lactose, it is your carbohydrate, so how it is, how it has to be broken down. When that capability is not there in the body, what will happen, right? So that is nothing but your body will show the intolerance. That is nothing but your lactose intolerance. So here you will clearly give the definition, you will give the underlying causes, and most importantly, you should mention it is a non-immunoresponsive, and you need to explain the structure of your lactose, and with completely the all your linkages, you need to explain the structure. This is since a very short answer diagrammatically. If you represent the lactose structure and these 2 points, if you give, definitely it will be a easiest. Then next is your hyperglycemia. Hyperglycemia, it is related to your blood glucose level. So that means what? Whenever your levels of your blood glucose are abnormal, that means they are above the normal range, then you will call it as your hyperglycemia. If they are below the normal range, you will call it as your hypoglycemia. So you just need to mention the reasons, the causes of the increase of your blood glucose levels, and you need to give a very short explanation of it. So I hope you're clear with this till now. So we have just had a brief brush on what are the contents that are there in your unit 1, digestion, absorption and metabolism of carbohydrates, and of course here the most important thing, the related disorders also, they will definitely pose 1 or 2 questions on this, and regulation of the blood glucose is for sure questions, and this, and then diabetes mellitus also, you can get it, you can expect this for your shorts as well as your very short answers, right? Then if you have seen, these are 2 important short notes that we have seen, and then we'll go to the next chapter, next unit, that is your unit 2, that is your lipids, that is which has most importantly fatty acids. Lipids, the first thing everyone, they are nowadays very much conscious, right, of the fats, the fatty foods that we are eating. Yes. So in this chapter, you will be completely learning about what are fatty acids, their definition, classification, and also the most importantly, definition, clinical significance of your MUFA and PUFA, right? Your polyunsaturated fatty acids, monounsaturated fatty acids, and most importantly, essential fatty acids, trans fatty acids, and digestion, absorption, and metabolism of lipids and related disorders, right? So how does see out of the among all the digestion process? See here we are like we completely don't maintain our lifestyles. We keep on dumping something or the other. Yes, our children morning we just have the breakfast, and after some time we will definitely put something else. We never give rest to our digestive system, right? How complicated the process of when we can constantly put on dump on things, and our stomach has to be needed rest, right? That's the reason earlier they used to put all the fastings and all so as to give rest to the digestive system, right? So this coming to the process of the digestion, carbohydrates, the digestion it begins in your mouth, and then coming to your proteins, it takes place in your stomach. Whereas your lipids, we all know very much that it's a complicated structure. The digestion and its absorption and the metabolism and related disorders, and they're quite complicated. But yes, once if you just understand the complete structures, it is very much easier. So you'll be learning about this, and you'll be learning about the compounds that are formed from the cholesterol. Cholesterol, yes, the first thing everyone, we are very much conscious about the cholesterol, right? Then ketone bodies, their names, their types, their significance, and lipoproteins, their types and functions. Most importantly, lipid profile. So, being a medical student, we should have a complete idea of what exactly is the lipid profile and what how we are supposed to see the parameters, and we should be in a position to judge by seeing the profile. And the next important one that is here, atherosclerosis. So these all are the contents that we see here. Then coming to your short notes. So here you'll be the most important question that is what are lipoproteins? You will just give brief introduction of what are, you'll give the definition of lipoproteins, like they are your combinations of your molecular complexes which contain your lipids as well as the proteins, and then you will explain their types, what are their types and their function in detail, and possibly give the diagram there so that the complete that will make appearance easier, I mean understandable and easier for you rather than giving a more theory of it, right? Then you will be just explaining in short of what are the types of it, and structure particularly you have to concentrate on the polar structure and as well as the non-polar structure. See, first thing that we know about the lipoproteins, they play a very important role in transfer of your lipids, particularly your cholesterol, the packaging, and then they play a very very important role, in they act like a substrate for the metabolism of the energy metabolism. For example, your tags, that is your triacylglycerols, whether you take your phospholipids, whether you take your cholesterols. So they are the precursors or starting materials for the synthesis of the various hormones. So if you just explain all this in all these functions and their types in detail, that should be fine with this question. Then the next one is difference between essential fatty acids and trans fatty acids. These questions are very much important questions. So these questions you would have been seeing in most of your earlier model papers and earlier examination papers also, you have seen these questions. So although this is a small one, but yes, it is a quite repetitive. Lipoproteins as well as your essential fatty acids and trans fatty acids. You see your essential fatty acids. Essential fatty acids, with the term only we can tell essential, they are very much required, but they are not synthesized in our body. So we require them through our food or diet. And trans fatty acids, the first thing that we get in our mind, trans fatty acids, that is the one which is not required for us. This has been prepared because of the hydrogenation, right? Because of the complete hydrogenation process. Just give a 3 to 4 point difference between your essential fatty acids and trans fatty acids. So this is also an important question. I hope you are understanding, and I hope the session is engaging for you all. And the next, coming to your next question, next session, that is your very short answers. Very short answers. Coming to the ketone bodies, they are very much important. Ketone bodies, you need to just explain what are the various types of your ketone bodies, the important ketone bodies that you have in your body. You need to explain about them in short. You need not give a complete thing, but you need to just highlight and underline the important terms. Like for example, your ketone bodies, they are nothing but your metabolic products. They are the metabolic products that are produced in excess in our body. right? So because of they are produced because of the breakdown of the fatty acids. So once your fatty acids are broken down, then your ketone bodies are being produced, and they are the alternate source for your glucose. So during our fasting times, so still we are able to maintain. Say one day we will be fasting, 2 days we'll be fasting, but still we can survive, right? That's just because of this ketone bodies, right? So yes, definitely we have a psychological feeling that we didn't had food, so I'm okay, I'm tired. But actually we can starve even for more than 10 days because our body will definitely have this reserves, right? So you need to explain what are the types of the ketone bodies. Like for example, you need to two main. You need to explain about the acetoacetate and also your beta hydroxybutyrate, right? Then the next one is your what are fatty acids. You need to just give the definition of your fatty acids, how they are your aliphatic hydrocarbons, and then how your what are their the structures. You need to explain it, right? The next question is about your normal blood levels of your cholesterol and your urea. You need to just give the levels and you need to just explain about this. So I hope you're clear with this, you're understanding, right? Please do subscribe our channel in my Prakara. Now, coming to the third unit, that is your proteins, wherein here you will be learning about completely the classification of your amino acids. Amino acids, they are the building blocks of your proteins, right? A complete human body is made up of your proteins, right? All your cell signaling molecules, your major DNA structures, you will also find content of the proteins there, right? So all your complete protein build structure and how the complete three-dimensional structure and four-dimensional structure of your proteins are being formed. So here you will be learning in detail, first basically about your classification of your amino acids based on the nutrition aspects and their metabolic rates with their examples, and you'll be learning about the digestion, absorption, metabolism of protein and its related disorders. Most importantly, your proteinuria, that is for sure short, you'll be getting this question. Then you'll be learning about biologically important compounds that are synthesized from the various amino acids. See, as I said, you from this amino acids only the proteins are being synthesized. We require proteins in our body for each and every activity, each and every metabolic activity. So what all are they? The only names you'll be learning on this. And of course, inborn errors of the amino acid metabolism, particularly related to the aromatic amino acids, we'll be learning in brief in this chapter. So if you see here, this is the plasma protein. You'll be learning about plasma protein and you'll be learning about its types, its functions, its normal values, and of course the causes of the proteinuria and your hypoproteinemia, hypergamma-globulinemia. All this you'll be learning in this chapter, and most importantly, the highest weightage, that is the most shortly you'll be getting this question: principle of electrophoresis, normal and abnormal patterns of your electrophoresis. See, we coming to the first thing, what we'll get it in our mind: electrophoresis. Electrophoresis, that is movement of the molecules based on the size and charge. So here you will be learning how your protein molecules can be separated, how you can test your the globulin levels, whether they are normal or abnormal. And of course, in this chapter, you'll be learning about the various electrophoretic patterns in brief. Right? So the important questions of this is firstly definition of your amino acids. What are your amino acids? And then how the digestion, absorption, and metabolism of protein will take place. Right? If you see the digestion of your proteins, it takes place in your stomach. Coming to your carbohydrates, it takes place in your mouth, whereas your protein digestion, it takes place in your stomach. Stomach because of your dilute HCl, and that is your gastric juice that is present. So in brief, you will explain the process diagrammatically. So you need to just define what is an amino acid, and then you need to explain the digestion process, absorption with the separate headings, and the metabolism and its related disorders. Then next is here how you will explain about the essential amino acids. See how we have your essential fatty acids, even we will also have your essential amino acids. Essential amino acids means what? They are essential for our body, but we cannot synthesize it. Right? Out of which we have 9 essential amino acids, and the easiest way to remember that is mat, will, fly. That is Math will fly. So M stands for your, the important one, that is your, you can just methionine, your alanine, then your threonine, your valine, isoleucine, leucine, phenylalanine, right? And this is how you can easily remember this. That is your histidine, right? Mat will fly, PHLY. So H stands for your histidine. So this is the easiest way to remember your essential amino acid. That is your histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. How is the easiest way? Mat will fly. M stands for your methionine, A stands for your alanine, T stands for your tryptophan, V stands for your valine, I stands for your isoleucine, L stands for your leucine, then P stands for your phenylalanine, H stands for your completely your histidine, and then Y. So valine. So in this way, you can easily remember all of your essential amino acids. So wherein they are very much important for us, but we don't get from a in our they're not synthesized in our body, but we should get it from our food. So these are the two important questions which have shortly been asked in earlier question papers. Then if you see, you'll be next question is what are aromatic amino acids? You need to just give their definition and the structure. In the sense, just simple structure. The two main important aromatic amino acids for us are your phenylalanine, tryptophan. The other one is your tyrosine. So these three, if you just explain its structure, draw its structure, and then you just tell it, define it, that should be fine. The next question is explain electrophoresis, the principle behind it. So how the particles will migrate in presence of how your DNA particles or your protein molecules, how they move, when they are placed in a gel. So all the principle behind it, based on the size and the charge. When you explain this in short, that should be fine. The next question is what are the causes of your proteinuria? So what is proteinuria? What are the major causes of it? When you just give a brief explanation, proteinuria, that means what the abnormal level of your proteins in the urine. Proteinuria. So the brief causes few 2 to 3 important points when you explain, that should be sufficient for this chapter. I hope you're understanding that, and I hope it is clear and this session is helping for you. Please do subscribe our channel. Now comes the most important chapter, that is your Unit 4, Clinical Enzymology. So here it is, in this chapter you will be learning about isoenzymes, their definition and properties, and enzymes of your diagnostic importance. And also you will be learning about the various liver diseases, which are caused, why are they caused, and also the most importantly, you'll be learning about the enzymes, that is ALT, AST, ALP, GGT, and you'll be learning about myocardial infarction and what are the cardiac troponins into it. And of course, you'll be learning about the muscle disorders and the enzymes related to this, the mechanism behind this, bone diseases, that is your ALP, prostate cancers, and you'll be learning all this in this particular chapter. If you see here, just what are isoenzymes, you need to just give the brief definition of it, and you need to explain how does the competitive inhibition of this enzymes takes place. In short, with the diagrams when you explain, that will be very much helpful. The next is here, explain about the various factors influencing the enzyme activity. So enzyme, definitely they are proteinaceous in nature, and there are various factors with which the enzyme is being affected. Substrate concentration, it can be the pH, temperature, and this way the various 5 important factors that are responsible. So you'll briefly explain it, and with the graphs you will completely show, okay, these are the factors that are responsible, right? Then the next question, it is about, you will be learning the important one is it's your holoenzymes and apoenzymes. Even this also, if you can just explain with the structure, what is a holoenzyme and what is an apoenzyme, and which part of it is playing an active role. And then if few lines, if you just explain about this, that should be fine with this. The next is here enzyme inhibition. Inhibition is to stop. So how does this enzyme inhibition takes place? In short, if you need to explain about this, and 3 enzymes of the diagnostic importance. enzymes. Any three enzymes of the diagnostic importance, which you need to explain about it, which you have seen in the earlier thing, in the earlier slide. It can be your ALT, AST, ALP, which are diagnostically they have the value, and definitely it is like they are responsible for some sort of disease or some if their abnormal levels will definitely responsible for some sort of disease. So those you will be learning. They are of diagnostic importance, right? So next, we'll be going ahead with the unit 5. This is acid-base maintenance, wherein you'll be learning about what is pH definition, which is nothing but the negative logarithm of the hydrogen ion concentrations. So how does the pH plays a very important role in the biological systems? This regulation of your blood pH, blood buffer, how does it takes place respiratory and also in the renal levels, and your ABG, what are the normal values of it? You will be learning about this. And acid-base disorders, their types, definitions, and causes. Like what are your various acid-base disorders you will be learning in this chapter. And out in this chapter, the most important thing is renal regulation of your acid balance is very much important question. Here in this, you need to diagrammatically explain the complete structure, complete mechanism, maintenance of your H plus ions, how it is taking place, how your bicarbonate ions, the regulation is taking place. You need to explain about this in detail, right? Then if you see, next coming to your question, normal range of your random blood glucose, your serum cholesterol, serum bilirubin. In this, you need to just explain what is it and what are their normal values. With that, it would be easiest for you to write down rather than giving the big answers, right? Then next, if you see what are the functions of your ascorbic acid, you need to give 5 important functions of it. So for very short answers, that should be fine. Then the next important one is your blood buffers, right? Then if you see the next chapter, that is your heme catabolism. In this, the most important, your heme degradation pathway and how does the jaundice and the various types of your jaundice causes, and urine and blood investigation, that most important test, Vandenberg test. How is this test done? And what are the how do you do this particular test? What are the parameters for it? You'll be learning in this chapter, heme catabolism, right? So the important questions for this chapter, you see here heme pathway in detail when you explain it diagrammatically, and you should, next question is you have to give and list the uses of what is the use of your Vandenberg test, right? I hope you are understanding this session, and I hope you're not getting any doubts. I hope you're clear with this, right? So next, let us go ahead with the other chapter, the 7th chapter, that is your organ function tests. So we will be in this particular chapter. So what are the functions of that particular organs and related functional tests related to that particular organs. So if you see your renal test, your liver test, RFTs, liver function test, LFT, your thyroid test. Yes, nowadays we are very much aware of all this. Everyone, any common man is very much aware of this. Coming to the medical students, this chapter has very good importance, right? If you see here in this particular chapter, next important questions, normal levels. So in everywhere, the levels are to be learned, they have to be remembered and they have to be learned in the easiest way. Then if you see your sodium levels, potassium levels, calcium levels, and the heat coagulation test. The second one is your heat coagulation test. It is a first short question, so important question. And above that, the most important one is your RFT as well as your LFT test. So you need to just give the definitions whenever you get in your short answers. Then the next chapter, that is your immunochemistry, wherein you'll be learning about the structure of your immunoglobulin and how your most important test, ELISA, how it is being done, and how you have to interpret the test. You'll be learning in this chapter, immunochemistry. You see the structure and function of your immunoglobulin. You need to describe the complete 5 different structures of your immunoglobulin and then Y-shaped model, and also clearly you need to mention its functions with each and every immunoglobulin. So diagrammatically, you need to give that and you need to explain about this in this. Then coming to your explain about immunoglobulin G, here you will be explaining about the structure and its functions in this chapter. Then next, if you see here, very short questions, advantages of your ELISA test, and This important question is your structure and function of your immunoglobulin. It is first short question. It was being asked in earlier question papers, and most commonly asked question, and it's a repetitive question. Then next, if you see around, write the types of the immunoglobulins, wherein you need not elaborate and write, you need to just mention what are the types of your immunoglobulins. So let us go to the next session. So the important questions, we just had a brief intro and brief overview of all the syllabus, the important chapters, and individually the questions that are being generally asked or repetitively asked questions we have gone through, right? Now we'll be going through the important questions. So in total, if you see 5 marks questions, that is your short questions of your 5 marks. So in these questions, for example, here if you see the structure and functions of your RNA and DNA, so clearly you need to draw the structure of your RNA, the loop model structures, and also your DNA structure, and differentiate with the functions. In short, when you draw the diagram and you give the differences, that should be fine for this question. Then second one is describe the reaction in the synthesis of urea. So you need to just explain whatever the reactions that are taking place in the synthesis of your urea. And then the next question, this is the most important urea related question, it's very much important. And afterwards, the next question is, what do you mean by lipids? So you need to give the definition of it, and then you need to explain the various types and uses of your fatty acids. Then this, of course, as I was telling you, the composition of the carbohydrates, glycolysis. Glycolysis with the pathway and with the general net equation, that should be fine for this 5 marks answer. The next question that is here, we need to write in detail the chemistry of vitamin D and its dietary sources. So you need to write, you need to give the chemistry of your vitamin D and what are the dietary sources, that is from which diet you're getting sorry, your functions and the deficiency disorders. The next important question that is here, we'll be learning about the beta oxidation of the fatty acids, palmitic acid oxidation, right? Now the next important sorry students for the inconvenience. So if you see next important 2 marks questions, blood buffers, they are very much important. Active transport. In this active transport, you need to mention what are the transporter. You need to just give 2 marks. It's a short answer. You need to just give the definition of the active transport. The third important question, write the steps involved in the Benedict's test. So you need to write what is the procedure behind your Benedict's test and what is the main purpose of it, right? Then the next one is your sodium pump. We need to define the sodium pump and its role. And of course, you have the antioxidants, diffusion osmosis. You need to give the difference between it. The ELISA test, functions of cell, electrophoresis. You have your phenylketonuria. This is very much important question, and for sure question this is. You need to explain what is it, what are its causes, right, and the treatment for it. Then anion gap, it is very much important. Acid-base balance, urea cycle for 2 marks. You can just give the brief outline of it. Gout, isoenzymes, polysaccharides, your gluconeogenesis, and your regulation of your blood glucose level. This question has been asked in n number of times. If you see, it was repetitive question. It's a very important question. The next is your HMP pathway. And of course, triacylglycerol synthesis in brief, if you can explain for 2 marks, that should be fine. Then next is your Benedict's test. So these are your transamination, metabolic alkalosis, Rothera test, ketone bodies, and important bile salts and their function. Deamination, biochemical function of your calciums, Golgi apparatus. These are the most important questions which are frequently being asked, and this can be explained in short. I hope you are understanding and you are clear with this, and I hope there are no questions or queries related to this. So if there is anything, please post it up, and please don't forget to subscribe our channel. Now, here comes the most important thing. You need to have a complete idea of how our exam model paper is. So, based on it, you can have your own preparations. Now, you have understood out of this complete 8 chapters, the weighted chapters, you are very much clear. Now we will have a complete look around with the exam model paper. How does your exam, your degree, how does it come for you, and what all are the important uh, aspects in this, like what are the marking scheme and all. We will have a complete go through of it. You see here, this maximum marks is 75 marks, and here you look over the first part, first section, you'll be seeing that you need to write short notes. Short notes, that is almost, it is like you need to write any three, and each carries here 3 into 5, that is your 15 marks. So here if you see again the repetitive question, your oral glucose tolerance test, its procedure, and in brief you will be drawing the curves, types of curves of it. The disorders of your acid base, you need to just explain what are the disorders related to your acids, and what are the disorders related to your base, right? Then the next one, if you see your lipoproteins, this is also a very much important question. If you have seen these questions, we have already discussed under the important questions. Isoenzymes and their properties individually you are supposed to learn. Then if you see immunoglobulin structure as well as the function, diagrammatic presentation, and then its function. Then next comes your important, write a very short note, answers on all of the following. So here you need to name the liver enzymes, and you need to write the normal values of your ABG and the causes of your proteinuria. So in these three questions, it is like very short answers. So they are carrying 2 marks. So 2 marks questions, we need to just give a brief of it. That's it. We need not explain much. Then if you see your choose the correct answer questions, usually these choose the correct answers or your MCQs, which is like 4 into 1, 4 marks. They will just go with the values which are being the parameters that you have been learning in the syllabus. So from there, usually you will get this sort of questions. Like if you see the saturation values of your hyperbilirubinemia, how it is caused. If you see a lock and key model, what it is known as, right? So like this, the one which are usually they will cover from the parameters, and this completely lock and key model is an important question. And then the, of course, the factors that are responsible for the denaturation of proteins. We know very well what is the denaturation of the protein. Denaturation, it is nothing but the breaking down the structure of your protein. So what are the factors that are not responsible? They have asked into this. So you need to check this, and we need to answer about this. Then if you see, when all the three next question, they have been asking us about the pH value of the three values of your pH, your bicarbonates and carbon dioxide are abnormal. Then what is going to happen? So this 4 questions are being posed into this. So this is how we are having our model paper. We see completely the first session, you'll be learning about 3 into 5, if you see here. In the first session, you'll be learning completely, you'll be writing the short notes on these particular questions, and that is 3 into 5, which is 15 marks. Then again, if you see the second aspect, that is your very short answers, this is almost 3 into 2, 6 marks, and then your MCQs, which is for the 14 marks you'll be learning. Basically, in this way, your pattern will be, your question paper pattern is as such. So if we go back to our important questions, if you see over here, I'll just give a quick repeat of the important questions. If you see here, the structure and functions of your RNA and DNA, and you need to describe here the synthesis and the reactions of your urea and urea cycle. These questions are short questions, are the most important questions that are being asked. And of course, the composition of your carbohydrates and your glycolysis, all your pathways are very much important. You need to concentrate on those questions. And the weightage units, as we have seen in the before slide, when we have learned in our first slide, if you see here, weightage units, your carbohydrates, your proteins, your clinical enzymology, acid-base maintenance, heme catabolism, immunochemistry. So most of the questions are being repeated from this. So if you would have seen complete contents also, you will understand like how the questions are being repeated, right? Yes. So this is all about your complete weightage units. Out of your 8 units, that is, you have your carbohydrates, you have your proteins, and you have the clinical enzymology, acid-base maintenance, heme catabolism, immunochemistry. These all are the weightage chapters and are very much important. Particularly carbohydrates, the classification of your carbohydrates, and your carbohydrate metabolism. All your pathways, they play a very important role, and those questions are being asked much frequently based on their importance, right? So any queries, you can just ping on to me. You can drop down your message. So this is how these all are the units. I hope you are clear with this, right? So this is all again, you just go back the important questions, very short answers, and of your first unit. And if you see your lipids also, the important questions, your lipoproteins, your essential fatty acids, trans fatty acids, they are very much constantly or repetitively asked questions, right? Then in your third chapter, that is your amino acids, you will be the most important concept, that is the digestion, absorption, and metabolism and related disorders. You need to concentrate more on this because more sort of questions will be posed on this particular topic. And of course, essential amino acids, electrophoresis, this technique also, this is very much important. And the weightage units, these all are the weightage topics. So I hope you are understanding and the session is comfortable for you. So this is how a complete view of all the important questions. This is what you are having your exam model paper. So children, students, so I hope you are understanding the importance of what the weightage units, and you're understanding what I'm trying to tell you people. And if there is anything, you can just put down the message. So now coming to the last aspect of our thing, please don't forget to subscribe our channel, and I hope I'm making you understand. I made you the topics clear. So here we get you with a new concept, new thing. That is our 1 year curriculum course. That is your mini learning project, wherein this particular project you can access with just 149 rupees. Just 149 rupees. Just imagine. So in this, with this, you are the important features of this project. It is like you will be provided with the various video lectures, with the student notes, important key terminologies, and of course self-assessment. The content, we get it everywhere. We get the content over the net, but what to read, how much to read, and what to be presented, and what are the important aspects of it, like what and all the notes, the short notes, what and all the points you need to cover, and important key terminology. Most importantly, the self-assessment. The self-assessment, how are we going to assess ourselves? So this is very much important. So these all we will be, you'll be getting just for just 149, right? So what do we get for 149, right, nowadays? So for 149 here, we are getting this package which is valid for 21 days. And I hope you're enjoying this session. And yes, definitely this MLP will definitely help you and with the good video lectures, student notes, key terms, and self-assessment, right? So you can just, for more details, further details, you can just go on to the number given below. And of course, you can ping us over this particular website. You can access to the website here, right? So here in this, you will definitely get a easy understanding video lectures, complete student notes, key terms, and of course, self-assessment. Self-assessment is very much important for us, right? So please do join into this particular course, and I'm sure definitely it will help you out, right? What are we getting for just 149, right? Right? So this is what we are getting for 149. Video lectures, student notes, key terms, self-assessment, right? So I hope children you are understanding it, and you are very much clear with the syllabus. You are very much clear with the content, right? So you have understood what all are the important questions, on what topics we are supposed to concentrate, right? Yes. So yes, here is again back for you this BSc Nursing course, first year curriculum MLP, wherein here mini learning project It is for each semester. It is almost valid for 21 days trial pack. You can just have the trial pack. This trial pack is valid for 21 days and it's just for 149. Wherein the important features are you'll be learning about the video lectures, you'll be having your student notes, easiest notes. Easily you can learn it and you can present it. Key terminology, what terminology we need to concentrate, and if what terms are being posed, that will be very much easier for the to score the marks, and of course most importantly the self-assessment, right? So yes, children, I hope you are clear with the sessions. So anything else you would like to ask, you can just drop down your message. Okay, well, I hope you are enjoying the session, you are understanding what I'm explaining, and I hope it has been benefited for you, right? I hope that would have been benefited for you, and you would have gone through the important questions, and of course, all the details are being given here, and I hope it is very much clear for you. So again, just let us have a look over to the first slide. We will just look over to the complete important questions. If you see, these all are the important questions that we are discussing, that we have discussed how to write the 5 marks questions, and how to write your 2 marks questions. In short, how we are supposed to briefly put those questions, right? Yes. So these all are the most frequently asked questions and important questions. Right. Well, so this is how we are presenting you all with our new project. That is your mini learning project, which is almost, it is for rupees 149. It is valid for 21 days. It is a 21 days trial pack, right? So just imagine students, like we are getting in such a 149, we are getting all these features. You're getting your video lectures, student notes, important key terminology, and of course self-assessment. Well, students, I hope you're clear with the content, and you're clear with the topics that we have discussed, the important questions. Then do definitely subscribe to our YouTube channel, and definitely try on just experience our mini learning project. Thank you so much. So let me sign off.